All right, welcome everybody. Good morning, afternoon, and evening. Again, thank you for joining us at the Ask Us Anything panel discussion presented by the top PBPK and PBBM scientists with over 20 years of experience. A few housekeeping notes today. For optimal video and audio connection, we highly recommend you close out any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously. By attending this call or participating in the Q&A session, you are allowing us to contact you for follow-up. This call is being recorded for future playback. Today's session will be broken into pre-submitted questions, fun trivia questions for the audience, some poll questions, and your live questions. To submit your inquiries, please use the question pane in your control panel. If you're experiencing technical issues with audio or video at any time, please raise your hand or send a message to me, the organizer, via your chat screen. Now, to begin, we've got a poll question. How would you classify your experience with GastroPlus software? We'll give you a few seconds to answer. If you've joined us earlier this week, you've seen this question more than once. So either beginner, intermediate, advanced, or if you have not used, please make your selection now. All right, just a couple more seconds before we get started. Thank you everyone for participating. And it looks like we've got a pretty good mix this morning. So it is my pleasure to now introduce one of today's moderators, Mr. Dan O'Connor, Director of Business Development for Simulation Sciences. Go ahead, Dan.
Thank you very much, Arlene, and welcome to the folks that have joined us on the last day of the week here for the Ask Us Anything panel. As many of you know, we're now all pretty much working from home. So if you do hear my two dogs in the background, they are looking to voice themselves. So I'll apologize in advance for any noise that you hear with them barking for the moment. But I did want to, again, invite you to join the GastroPlus user group. We found that people really do learn an awful lot. They are able to share with both techniques and tips and tricks on how to better employ not only the ADRs, the additional dosage routes that you've come to learn a little bit about this past week, but other variants of how to best approach your research. It's my pleasure this morning to talk a little bit about who will be on the panel. I'm joined by John DeBella, who's the president of the Lancaster division of Simulations Plus as a co-moderator. But I thought I'd just take you through a brief introduction of each one of the panelists here by switching my screen. Hopefully you can see that, Arlene, yes? I'll say that's yes.
I can see it.
Okay, great, thank you. All right, the cavalcade of stars this morning, afternoon, and evening are including Mike Bolger, who's our chief scientist, Vera Lukachova, who's director of simulation sciences, John DeBella, who, as I mentioned moments ago, will be co-moderating and is the president of the Lancaster division. A recent addition to our team, Sandra Suarez-Sharp, who's a vice president of regulatory affairs, Grace Frakowitz, team leader of the Simulation Studies Group. And there's extra credit if you can spell her name. That's what she used to do when she was a teacher. Jim Mullen, senior principal scientist. Kay Zito, who's a senior scientist. Maxime Lamerdi, who is our scientist in Switzerland. And we're also joined this morning by Jessica Spires, who doesn't yet have a picture. I don't know why, Jessica, but that's the reason that she doesn't appear here. So with that, I'm going to turn it over to John to offer the first comments and maybe a question. John?
Dan, thank you very much. First, hello, everybody from rainy Los Angeles, California. Weather for the last month or so has been nothing but sunshine. temperatures in the mid to upper 70s. And it seems today the universe has decided that it's a good opportunity for me to get cozy with a cup of coffee and have a nice dialogue with my colleagues from Simulations Plus and a number of users out there globally in the world. I think, Dan, would you say overall this week of workshop from home lectures has been pretty successful?
Outstanding, as a matter of fact. The response has been fantastic. I think Arlene shared with us that there were as many as 858 registered folks for the seminar series. We did have one glitch this past week, and I did want to mention that we will be having our dermal ADR presentation next Wednesday, and I'm confident that Arlene will talk about that at the end. But yeah, no, it's been outstanding. Lots of questions that have come in after the presentations directly to me and to the other scientists that have presented. So hopefully we can find future topics that might be of equal interest to our users.
Yeah, yeah. You know, transitioning over to the theme for today, Ask Us Anything. You know, as you alluded, there's been a lot of questions that have been asked on all of the various delivery route models that we have inside of GastroPlus. There is, though, I think a pretty common theme for a lot of these, and that is, you know, our abilities to take in vitro information and have some of that be translated into the in vivo environment, and you know, then potentially utilizing both what we've generated in the lab and what we've generated in some of our preclinical or clinical studies to build and form in vitro in vivo relationships. And so I think as part of, we could call this an initiation of sorts, I'll call on our newest member of the Simulations Plus team, Dr. Sandra Suarez-Sharp, who was with the FDA for many, many years and now serves in the role of Vice President of Regulatory Affairs, to answer a question that, again, I think is kind of appropriate and covers an overarching theme. So Sandra, can you maybe spend a little bit of time talking about how we might be able to build in vitro, in vivo relationships, and, you know, regardless of the delivery route, really, what sort of data do we need to have? Can we do this with just a single set of in vitro dissolution kinetics or in vitro phys-chem characterization and corresponding in vivo data, or is more needed? And let's not forget for the panelists to make sure you're unmuted.
Can you hear me now?
We can.
Thank you, John and Dan, for the nice introduction. It's really a great pleasure to be here today, being part of this panel discussion and seeing the interest in the utilization of physiologically based modeling, especially GastroPlus, in answering your questions and your regulatory questions or drug product development questions. In terms of your question, John, I mentioned a really good question that we saw it when I was at the FDA. People really are not making sure what is the difference between IBIVC and IBIVR, especially when it comes to regulatory application. And I would just let me spend a minute trying to define it. And I would say that in general, we can say that an IVIVR is a failed IBIVC. That means then that the data that was used to develop that IBIVC then is relevant, can still be used to make, I would say, regulatory claims. Now, data, you talk about which data is needed to build this IBIVR, and as I mentioned, the data is pretty much similar to what is being used to build an IBIVC. Then it means that we need to have a rank order relationship data, meaning changes data that comes from changes in the formulation that has been evaluated via dissolution, and of course in systemic exposure. So the bottom line is that to build an IVIVR, we need at least 2 sets of data, meaning data from 2 formulation variants, so that we can really define that relationship. Again, the relationship between, if we were to change the critical material attributes and process parameters, how is the dissolution is going to be affected, and at the same time, how is the systemic exposure is going to be affected. So again, hopefully it makes sense that to build that relationship, we need at least 2 sets of data. And hopefully that answers the question, John.
I think so, very well. Would it be, in your opinion, that internally, at least for R&D purposes, to make some decisions, there is still value in trying to leverage even just a single set of data at an early stage, if that's all you have available?
But I would say so, but you could use it. You would try to build a model with that, and use the software to make predictions on what would be a potential systemic exposure if you change the in vitro release rate. But still, I mean, and again, yes, I mean, the short answer is yes, it's valuable, but from regulatory perspective, I would say that it will make sense, hopefully, that we'll need more data to gain confidence in using that relationship for regulatory decision making.
I think we all appreciate that those comments, because there is sometimes a misperception on what it is we could potentially do to try and inform regulatory decision making. And there are a lot of opportunities available, but we do have to make sure that we've built up enough confidence in our approaches and have validated and verified our predictions with multiple data sets. If I try and build off a little bit from that question and the response from Sandra, Jim, welcome to the hot seat. There have been a number of questions that we didn't get a chance to answer following Monday's webinar when we focused on pulmonary absorption modeling with the PCAT model. And one of the questions asked was, what are our experiences using some of the theoretical deposition calculation approaches, ICRP 66 or Finlay methods, versus trying to use experimental data directly, whether it's from ACI or NGI studies. Is there a preference for one approach versus another when we're trying to build up the PCAT model and do some simulations?
Yeah, I mean, so as I'm sure everybody knows out there, the devices used to deliver dry powder dosage forms can all give, different deposition profiles based on how efficient they are and how easy they are for the patient to use. So the ICRP method will get you somewhere in the ballpark, but you may need to actually use some in vitro data to define some of the inputs at certain times. I think the best thing you could probably do, though, is you'll first maybe need to create some sort of a correlation between let's just say the NGI, for instance, how much is deposited in the different sections of the NGI versus, maybe some real deposition data, if you can afford to get that. If you can create some kind of in vitro to in vivo correlation with the deposition measurements that you're doing in the NGI in the lab, that makes the translation to GastroPlus, I think, the most optimum. So I think trying to come up with some scaling to get the NGI directly into GastroPlus is what maybe the companies should be focusing on. If you use the NGI directly, without some sort of a correlation in between that, I'm sure that'll get you, it's been our experience that that'll get you in the ballpark as well. But I think fine tuning that with some like intermediate correlation between the two, I think is something that would be a good idea in most cases. I think that answers the question.
Yeah, very nicely. And stay on that hot seat, because as kind of a follow-up and related to that, in the example that you went through on Monday for tobramycin, the idea was for us to try and take some of that measured deposition fraction data and then keep that fixed and utilize whatever available PK data we have to optimize parameters more focused on either permeability or absorption rates to get a model that better matches with what we've observed. Is that a general approach that we've taken with the pulmonary model? Is, you know, when we have confidence in the deposition fraction data, we then shift our focus towards evaluating some of the unknown or uncertainty around permeability or absorption rates into the systemic circulation to capture the PK data?
Yeah, correct. Yeah, exactly. And the point is that you don't want to globally fit all your data, right? Then the model's not really useful. You're just fitting data, right? So what we had done in that example was to take one of the data sets to train sort of the absorption part and focusing on that. And obviously tobramycin's a very low permeability compound, so that is the most sensitive parameter that's most likely to be causing the, you know, deviation from your experimental data. So that's what we had adjusted to train it against that one data set for the pulmosphere. Then we want to fix that for new formulations. We want to try to then use that to predict other formulations to show that the model is you know, useful and robust in predicting other conditions, maybe other particle sizes if you use the deposition model, you know, any other new doses, dose levels. So basically the approach is to use a data set to train the model, but then to predict after that, not to fit all your data sets. And I think that was the main point we were trying to make, and maybe I didn't very well explain that during the presentation, but that was the goal.
And you've done it just now, so we appreciate that. If we stay now on this theme a little bit of optimizing or fitting parameters, trying to build up a better model, Vieira, welcome to the hot seat. Lots of positive feedback on Wednesday's conversations around what we're doing for injectable products, and some questions that were asked are, you know, for the injectable formulations, you know, are there any critical parameters other than KP that we might want to consider optimizing or fitting in order to again better match with what we've observed either preclinically or clinically?
So that is going to depend on the formulation type that you are trying to simulate, right? With the solution data, and especially the examples that I was showing with midazolam and the doses that were used, it was administered as a solution. There wasn't much reason to think that precipitation would be causing any problems. So really the drug getting from the tissue into the systemic circulation or into the plasma was the most obvious process, and that is why we were looking at the KP, which would be driving that process. But for other types of formulations and compounds, you might want to look also at other parameters that are driving dissolution or possibly precipitation. You actually just recently worked on a compound where the administration was a solution, and the only way to actually match the observed PK data was trigger a significant precipitation and very slow redissolution of the precipitate. Okay? Well, it turns out that the model was right. The company actually looked at the data. This was in rats, and they found that, you know, on days after the injection, they started seeing issues at the injection site because the compound really did precipitate and started creating problems. So it really depends on the compound and formulation type that you are looking at.
That's very helpful, Vera. And sticking with this theme of formulations and compounds that we're evaluating, and making sure that we kind of have this good general understanding of some of the mechanisms that we have pre-built into the models, but maybe some others that we would want to consider. This is a question for Jessica, and unfortunately, we had some technical difficulties on Tuesday, and as Dan mentioned, the lecture on dermal absorption modeling has been rescheduled for next week. I know there's a lot of demand to learn more on that topic. Maybe to give a little bit of a teaser of what is to come next week, Jessica, some questions about the mechanisms for the dermal model, and are we able to capture the impact or effect of permeation enhancers for improved absorption with the TCAT model?
Yeah, that's a very good question, and it's one that we get a lot. So there are some limitations here, and that's primarily that currently we cannot predict the effects of permeation enhancers. If you say, okay, this is my formulation, this is how much of this permeation enhancer we have in it. Currently, we don't have models developed that will give you a prediction of how much to what extent the permeation will be enhanced. However, what you can do with the model, and what you could do if you had enough data, would be to build your own correlation, is that you can adjust the permeability in the skin layers. So, if you know that there's a permeation enhancer present, we have predictions for kind of the basic permeability of the stratum corneum. And you can say, okay, well, I know that now that I'm including my permeation enhancer, this can have the effect of increasing diffusivity, and or it may have the effect of decreasing the partition coefficient. So, quantitatively, we don't necessarily know exactly where it is, but qualitatively, we know what direction to move in order to enhance the permeability of the stratum corneum. So, you would need some data to sort of calibrate the extent of the permeation enhancement. But once you had that type of information, you would be able to develop a model based on it. I would add also, obviously, this is something that we have a lot of interest in, and we're definitely looking into. This is primarily a limitation of data on our part to be able to build these types of models. So, as soon as we get enough information to start predicting the effects of permeation enhancers, we'll definitely include it.
Very nice, Jessica, and that is call out number one to the audience with regards to an expression of interest from our side to enter into collaborations whenever possible. We're always happy to engage in conversations and discussions on ways in which we could potentially add new functionality or make improvements to the software through partnerships with companies. For us, data is gold, and anytime we have an opportunity to work with data to build up some of that new knowledge, we're more than interested. Viera, we didn't get a chance this week to talk about the oral cavity model, but sticking with the theme of permeability, can we talk a little bit about the best ways of trying to define permeability coefficients in the oral cavity model?
Okay, so for this, I actually am going to show GastroPlus. Okay. So within the oral let me make sure I'm showing the right screen here. Okay. Within the oral cavity model, the diffusion, let me select one of the oral administration routes, oral cavity administration routes. And in this case, the absorption into the oral mucosa and diffusion through the oral mucosa is not driven by the intestinal permeability, but it is driven by diffusivity through the oral mucosa, through that tissue, and the binding in the tissue. Right? So these are the two parameters that you would be adjusting to calibrate the permeability through that oral mucosa and how the drug is getting into systemic circulation. The initial estimates that we provide are based on the log D of the compound. When we were developing first this module, we did have data, in vitro data through, I think, buccal and sublingual tissue for 8 or 9 compounds. And based on those, we were able to fit the FUTs and diffusivity through that oral mucosa tissue, and created a simple relationship for the FUT and diffusivity versus log D. So that is being used as an initial estimate, and you may have to further optimize or fit it against in vivo data in animals, or possibly in vitro data if you have measured the in vitro parameters. But these would be the ones that affect the diffusion through the or absorption through the oral mucosa.
Thank you very much, Vera, for the explanation and for teasing the oral cavity system. I promise to the audience we'll get a lecture on this model in particular here in the near future, so stay tuned for more information on that. Max, take us into intermission if we stick with this theme of permeability here for a little bit. Have we considered the concentration gradient of unbound, unionized fraction between adjacent ocular tissue and fluid compartments when we calculate passive permeabilities in the eye?
Thanks, John, for the question. So as I mentioned yesterday, I'm not sure exactly if we've considered that specifically, but the permeability that we have optimized in the eyes for different compounds were optimized on a set of, I think, 13 compounds with different status of ionization and also different protein binding in these different tissues. So probably the effect of ionization and binding to protein is lumped into the permeability that we have optimized. The issue is to obtain real measurement in vitro, because it's very hard to obtain like a retina permeability, for example. It's I don't think it's done anywhere. So it's very hard to really know the accuracy of all parameters. So it's something that could be looked at in more details, but it would probably require maybe some innovation regarding in vitro testing to actually obtain very real values. So far from what I've seen, I've only seen in vitro measurement for cornea, conjunctiva, and very few in sclera. So that's only 3 tissues out of the 10 or 11 that we have in the model.
Very nice, Max. And again, another call out potentially for any collaborations here on this front. Certainly interested in exploring ways in which we could take some of the data that is being generated and find better in vitro and vivo relationships. Arlene, we're about halfway through, and I think at this point we wanted to see if we have a lucky audience member who may be walking away today with a gift from Simulations Plus.
Yes, thank you, John. Okay, so tune your ears in and get your chat panels ready. To participate in this part of the trivia portion, you can win a $20 Amazon or Visa gift card. So we're going to read off a question, and then you're going to use your chat panel to submit and timestamp your answer. Winners will be contacted via the email that you registered for this webinar with, along with celebrated on our LinkedIn page, and you'll be emailed the digital gift card. Now, what happens if you cannot accept it? And I'm looking at you, FDA. I see some people in the audience. you can also donate that card. So don't worry, you can still participate and in the fun and compete with your peers. So to have your chat panel, it's the final pane down at the bottom in your control panel. You can answer to the entire audience, but the first answer, correct answer, will be the winner, in which I will contact you with your email to let you know that you've won and where to submit your gift card to. So here we go. How many different administration routes does GastroPlus include? A, 2, B, 4, C, 6, D, 8, or E, 10? And we can all thank Viera for asking this question. So please submit your chat response now. A2, B4, C6, D8, or E10. If you've been listening all week, how many different admin routes does GastroPlus include? We have our answer. The trivia question is now closed. And John or Dan, we'll toss it back to you for some more insightful questions.
I just want to say that I wasn't allowed to play, right, Arlene?
No, you were not allowed to play. Not today.
Rats. All right. Well, I do have a question that's just come in a little bit ago, and it might be one that's kind of for open discussion, because I think it may even push a little bit some of the technology that we have. But the question is, and I'll put this to the panel and let whomever wishes to respond. Can you predict the or if I could say the word, can you predict the toxicological and safety profile of a drug along with the PK parameters by GastroPlus?
This is Mike Bulger, and Dan, I would say the answer would be not necessarily with GastroPlus, although we can link in known toxicological mechanisms in the PD module to see what the formulation effects would be on various tox responses. But in ADMET Predictor, there's a whole host of toxicological predictions for mutagenesis, for H-erg binding, for a number of different endpoints, acute rat tox, etc. And we've actually participated in a number of the challenges coming from the NIEHS, Environmental Health NIH Institute, to submit models for various endpoints in the tox area. So if you're interested in the tox and safety profile of your new drug, check out ADMET Predictor standalone.
Thank you, Mike.
If I could add to that, the ADMET Predictor has variety of endpoints already. The other option would be looking more specifically at, let's say, toxicity in liver or in kidney in the near future, where the outputs from the GastroPlus simulation could be used in the DILIsym software, which is software produced by our sister division over in North Carolina, to start looking at the potential or start predicting the potential of the compound to cause the liver injury, or in the near future also kidney injury. Of course, that type of prediction would require some additional in vitro data, but through the use of combined use of those tools, it would be possible.
Dan, I just want to briefly mention, this is Sandra, that I've seen some very interesting papers published where, just like Mike was saying, ADMET Predictor has been used to determine the profile, systemic exposure of some impurities, for certain products. So yeah, it's widely used.
Well, that is the holy grail. I know, John, you've put forward the slide many times of connecting our platforms from ADMET Predictor through GastroPlus, through DILIsym, and now even with our sister division Monolix joining to kind of connect all of that. So, not so long in the distant future, maybe from very early discovery, both safety and toxicology information might be elucidated, right?
Oh yeah, absolutely. I think there's quite a bit of positive momentum, especially from our friends in the non-pharmaceutical space, to apply the tools almost in a reverse dosimetry way. Mike alluded to this in his response. There's a real desire to identify alternatives to animal testing, especially for chemicals, cosmetics, consumer goods. And so we've been seeing quite a bit of the use of the software where we might only have some very, very limited data available, and we want to try and get an early idea about what might be our safety margins if this chemical or this impurity or ingredient in a consumer household product were ingested or applied to the skin or inhaled. So there's already use cases that have been published, on that front, and there's this real desire to try and take whatever limited data we have and with some level of confidence or reliability, extrapolate that to different populations as well. And I think that would transition into maybe a next set of questions for the panel. I think we were asked some form of this question in just about every lecture this week. So I'll call first on Viera, but then also offer or invite the rest of the panel to also comment. Are we able to utilize these non-oral delivery models with the population simulator in GastroPlus? To what extent have we defined coefficients of variabilities around some of these inputs that would be specific to pulmonary, ocular, oral cavity, injectables, and so on?
So the short answer is yes. All of these administration routes are linked to the population simulator. In terms of the coefficients of variation, it's the same answer as for, let's say, any other physiological parameters. We do constantly keep looking through the literature, see what new information we can find, and if we can find enough information to define them based on known physiological variability, we do keep updating those in the program. Otherwise, we put in our best guess for the CV based on maybe variability in similar parameters and so on. So when it comes to CVs, we don't have those completely defined based on real physiological variability, but we do keep updating it as we go.
So you've alluded to that. Thank you, Vera. You've alluded to the flexibility that the platform offers. Jim, a question for you that was asked on day one. It seems there's interest. There has been some recent news about some of the treatments that are being evaluated for COVID-19 and the way in which this disease acts on the respiratory system. There's some companies, for example, Gilead, which are now evaluating inhaled product development work for some of their candidates. Are we able to create custom species models for pulmonary and really for any of the delivery routes? And if the answer is yes, what would be some of the parameters that we might want to modify or change or try and look up from literature?
Yeah, the model in a sense is completely customizable, right? You can change any of the tissue areas and whatnot. So if you have any sort of disease state, you can go in and sort of create that. Again, you will have to do some literature work to try to define that. We don't have any built-in disease models at this time, really, for the pulmonary model. So you'd have to go in and look at the effect of maybe the respiratory rate, the effect of deposition, you know, in a disease state patient, and maybe, you know, some of the lung is not accessible to particles and the drug anymore, so you might have to adjust maybe the size of the tissue and the area of the tissue, things like that. So these are what if scenarios that you can definitely test. You would have to define the physiology for that, and all the parameters in the model you can go in and adjust. So we do give that flexibility. It's just a matter of trying to determine good inputs for those different parameters. So yeah, those inputs are there. So yeah, you're freely able to customize.
Very good. And when we have that customized model in place, we can then make use of all of the functionality within GastroPlus, whether we want to do some population simulations for that species, whether we wanted to try to optimize doses for a particular product within that species.
Correct. Yes, the pulmonary model is linked to the population simulator. It's linked to the PSA. It's linked to the optimization routines. You know, it's set up to use even the multiple doses is set up in there. So it is really, you know, connected to all of the functionalities that we have in gastro. So you can, you know, do those types of different analyses.
Mm-hmm. Okay, good. We've spent a little bit of time today talking about permeability considerations in a couple of the different models. Maxime, if we do shift focus here for a second, with the eye model, with OCAT, where are we headed in terms of making some considerations for buffer strength or microclimate pH to help us calculate solubility or precipitation kinetics, and maybe the impact those parameters might have on mechanisms like tear fluid turnaround?
That's a great question. So actually, through our past collaboration with the FDA, we have developed a model that includes dynamic pH in the tears, where based on the tears production, the pH goes back to the physiological value. So we have that capacity already in-house, at least. And then it's a matter of how do we really calibrate that, because when we were doing that work, we couldn't find really much information in literature that could really correlate the tear production rate with the pH of the solution that would be used. And I guess this is then the third call for potential collaboration. Because if anyone has this kind of data, there could be a way. The code is basically ready to go. We have some couple of success stories, especially with the pilocarpine case study, where we were able to predict the increase in aqueous humor exposure based on the pH of the solution at the same dose. But it's just one case study, and there were not other really data available. So if anyone has some, feel free.
Thank you very much, Max. Mike, we haven't heard enough from you today. And kind of maybe piggybacking on what Maxime just responded to, there's been a number of questions that have been asked about precipitation kinetics. And I know that's an area that is near and dear to your heart. Are we able to utilize some of the more complex precipitation models that we've implemented in GastroPlus, where we've shown a lot of validation for oral delivery to the non-oral routes? And in your opinion, what would be some of the advantages to using something like mechanistic nucleation growth versus the first order approaches?
Okay, well, it's a good question. And when we take a look at our classical first order precipitation approach, or even utilization of multiple first order precipitation times, it's kind of a very empirical approach to modeling precipitation. It's pretty difficult to parameterize that from in vitro data. However, we do have a publication coming out, I hope fairly soon, on albendazole, where we did parameterize that using the transfer assay from Jennifer Dressman and Ed Koskowitz lab in Germany. So that's an interesting study coming up. But I would say the big benefit between first order precipitation and mechanistic nucleation and growth is that the equations that underlie either classical nucleation and growth or our mechanistic nucleation and growth derivation allow the molecule itself to define the level of supersaturation that is achieved and the size of the initial particles that are produced, as well as the growth of those particles and redissolution of the particles, so that you can easily go between doses where that supersaturation level changes due to increasing or decreasing dose, and the mass of the precipitate and the rate of production of the precipitate is accurately calculated by those equations. Whereas in the case of first order precipitation, a single precipitation time or multiple precipitation times would need to be fit to each single dose, because we know the equations define, not the first order equations, but the real nucleation and growth equations define the rate of precipitation to be changed depending on the level of supersaturation. So it is, you know, maybe a little bit more intimidating to go into that mechanism, and it's not something that you can use the optimizer for with the LIN4's derivation, but we think that with the newer derivation that Robert Franchkovitz has come up with, where the optimizer would just change the interfacial tension, that you could actually get some automated optimization of the components of precipitation.
Thank you very much, Mike. And I would say first plug for what we see here on this slide in the upper left corner, GPX. Not only some of the modifications that Robert has made to the underlying MNG model, but also improvements to the optimization algorithm itself should allow for, in this next version of the software, to do some, as Mike said, automated optimizations in a very efficient time frame. Arlene, there are still a few general questions that I'm happy to ask the panelists, but do we want to pause here for a poll question or another trivia question?
Yes, let's get one more question here for our audience. Based on what you've learned this week, what are your interests in using the GastroPlus ADR model? You can choose all that apply. We'll give just a couple of seconds here. Well, the answers are coming in. All right, a couple of seconds, and we're going to go ahead and close the poll now. So, some good responses. Support formulation development looks like it was #1. John and Dan, back to you.
John, I don't know, you said you had a few more questions. I was going to ask Sandra a question, but if you have one, please go forward.
You'll give me an opportunity to drink a little more coffee, Dan. Go for it.
Well, Sandra, this isn't a question that anyone asked specifically, but there's been a host of questions that typically ask in a general way. Given your experience, lengthy and expert experience with the world-class regulatory agency, FDA, You have about 150 world-class PBPK and PBBM scientists on this call. Can you just maybe share one or two key taking home, message for them in dealing with the regulatory agencies? Maybe as they're beginning the design or structuring of their research, what one or two pearls of wisdom might you wish to share with them?
Very interesting question, Dan. Yeah. So let me see. From regulatory perspective, I would say that what they need to do is, and regulatory perspective, and of course now that I've joined Simulations Plus and see the capabilities of the software and physiologically based modeling, I would say that they should, I suggest that they embark in implementing these tools to make better sense of the drug product. I think that definitely using these tools will help them accelerate their product. It will help them to make the right decisions. It will help them to reach to regulatory agencies and with the justifications of why they follow certain path. So I would say that as early as possible, they try to implement physiological based model into formulation development, into first in humans, into determining what would be the right dissolution method, into determining whether it would be certain doses appropriate or not for a certain clinical trial. So I would say that that I really encourage them to look into this.
Thank you very much. John?
Yeah, that was a good question, Dan. And, I'll take it from the other side. So maybe we're a little bit further down the line and thinking about how the models may inform regulatory decision-making, but going back a few steps. Viera, I'm going to call on you first. With great power comes great responsibility, but I also want to invite other folks on the panel to comment as well. As briefly as you can, because this is a question that really is something that we touch on in a multi-day, hands-on, intensive workshop. But if we're thinking about trying to develop or evaluate products that are going to be administered through non-oral routes, what would be a general strategy for building up that model? Do we try and start with IV oral data first and whatever in vitro information we have to then transition over to the delivery route of interest, or is there some other recommended method?
So the briefest answer would be yes. Yes, the general strategy would be, you just like with any other, formulation aspect, you do want to make sure that your PK is described correctly first, right? So ideally, you would be starting from IV dosage forms. If not, we've had success with, oftentimes calibrating the PK model first against oral administration, and then to again make sure that the systemic circulation is described correctly, and then start looking at the specific aspects that would be affecting the absorption and the formulation behavior with the specific administration route. I think that's the shortest answer I can possibly give.
It was so short, John's still on mute.
Well, I wanted to give a second to see if there was anyone else who wanted
anyone
to
else
comment.
on the panel? Sorry.
Yeah, Maxime here. I just think that rules may not apply for ocular, because if you're really focusing on the ocular drug penetration, unless you have some questions about potential toxicity and you want also to calibrate the non-productive absorption, maybe you can go straight with just ocular data, as we've done in a case study yesterday. So depending on your question, having the IV and maybe oral may not be necessary for ocular.
So that is actually a very good point, to add on to it, and that would actually be applicable with any of these routes, depending on whether you are looking or whether the compound actually is able to make it all the way to the systemic circulation, or whether you are designing that formulation more for local delivery, right? So, and with ocular, we see it more than with any others, but it could happen with some of the other routes as well, that the compound simply just won't make it past the injection point or past the administration point in which you would go directly to that specific local PK.
Yeah.
So having a brainstorming session with your colleagues and deciding as early as possible what you plan to do with that model, essentially fit for purpose approaches is kind of what we would recommend. And of course, making the best use of the data we have available. Even if it's limited, there's still a lot of really useful information we can get out of it at the beginning of a project. Sandra, there was a question from one of the from the answer that you had given towards the beginning of today's discussion. And actually, for my own benefit, I think it would be helpful if you could address it as well. Your thoughts on the difference in the definition of IV-IVR versus IV-IVC.
Yeah, so, in terms of purely definition, John, from a conceptual perspective, concept perspective, an IV-IVR, as I mentioned before, is a failed IV-IVC, meaning then that it has data there, as I mentioned, and you were mentioned, that can still utilize to make a decision, to help the regulatory agencies to make a decision. And so when it comes to its regulatory application, how do they differ? I would say that, as we all know, historically, IV-IVC is the gold standard into using this framework to support to be used as a surrogate for biowaivers. And IV-IVR still can, that data, as you were saying, it still can be leveraged into making decisions, especially when it comes to formulation development, formulation selection, whether certain specification is going to be of clinical relevance or not. But again, when it comes to making high-risk decisions, like if, for example, if a change in the formulation was a major, then whether or not we can utilize an IV-IVR to make that decision. I'm talking about IV-IVR developed based on physiologically conditions. Then it all depends on, the data and what justifications are being put forward to utilize that IV-IVR for the agencies to consider it as a surrogate for, or as a justification for a change. I'm talking about drug product quality change. So hopefully I was not too convoluted in giving your response, but they do have differences when it comes to especially this regulatory application. Excuse me.
Mm-hmm.
Sandra,
okay.
I appreciate that you made a clarification about regulatory versus making decisions in product formulation and in development, because I don't think of an IV-IVR as a failed IV-IVC, but I just think of it as a less expensive IV-IVC.
Right.
You don't have to buy 5 different biostudies and make 5 different in vitro release rates, but you can still learn a heck of a lot, and you can apply it to your generic formulation or your testing of product post-approval product scale-ups and other things. So they are valuable, and I think the main difference is just how much you want to invest in collecting the data to generate that validated IV-IVC.
Right, yeah, sure, and that concept, just hopefully I didn't confuse in my response, but that concept of leveraging that data use for within the IVIVC, which fails, still can be utilized to make decisions, to guide in many ways, including regulatory decisions. But again, all depends on what's the risk for that change.
Right, right.
Yeah.
Okay, well, I think we're at the end of our time. Arlene, is it coming back to you?
Yes, thank you, Dan. Thank you, John, and thank you to our panel. But most of all, thank you to our audience for attending all week and especially today. Please join us next Wednesday, June 10th for Dr. Jessica Spire's GastroPlus Transdermal webinar. You can register on our website at www.simulations-plus.com. You can look for all unanswered questions from today's session as well as all week on our LinkedIn page. And as a reminder, July is not only heating up with the weather, but we have more online learning events. DDD Plus, Membrane Plus, and PK Plus product workshops are also on the schedule. This does conclude our webinar for today. Thank you very much.